If DR doesn't work in humans, what else can we do? This is the topic of the final module in this course. In the last module, we have seen that CR extends lifespan and health span in laboratory animals, but the verdict on its effectiveness in humans is still out, and as we have seen, may not even be worth the trouble. So what other options do we have to live longer, healthier lives? Understanding the molecular and cellular events that drive aging, as we have seen in the second part of this course, are the basis for a whole plethora of anti-aging interventions that are currently under study in laboratories all across the globe. One of those interventions is as old as human storytelling. In Bram Stoker's Dracula and many other stories just like it, an old and dying vampire drinks the blood of a young woman and almost instantly rejuvenates to his prime. Of course, vampirism is not an acceptable way of rejuvenation, but there is some truth to these stories nonetheless. As we have seen earlier, somatic cells may become reprogrammed if exposed to the proper stem cell niche. This is the principle underlying rejuvenation by parabiosis, the surgical joining of the circulatory systems of two animals. If one animal is old and the other is young, the older animal gets rejuvenated, presumably via soluble factors in the blood that aid in stem cell reprogramming. Of course, that is not a very practical form of anti-aging, especially as the young animal is exposed to old molecules and will thus experience aging on its part. But one idea in the anti-aging field is to simply replace failing tissues and organs, and even failing cells. At the heart of this idea lies the so-called IPS, induced pluripotent stem cell. These are somatic cells that can be reprogrammed ex vivo into pluripotent stem cells. Once back in their primordial stem cell state, these cells would then be induced to differentiate into the desired cell type, which can then be used to either grow organs in vitro or to repopulate and rejuvenate failing organs in vivo. But what about more immediate interventions? By targeting molecular mechanisms of aging, researchers are hoping to mimic some natural mechanisms to elicit anti-aging effects, but hopefully without the detrimental consequences. Even though we know, however, a lot about the mechanisms of aging, we're still ways away from a practical drug. The development of any treatment for any disease is a long, drawn-out, and very costly process that only begins with the identification of molecular disease mechanisms and its molecular drug targets. After a small molecule shows promise in preclinical work, a lengthy set of clinical trials has to be performed that may take up to a decade to perform properly. These trials are to ensure efficacy and safety of the treatment. Only once 3 phases of trials have been completed can an application with the FDA be filed to bring the treatment to market. But a phase 3 clinical trial specifically designed for an anti-aging application would take more than a lifetime to complete. That's why most current so-called anti-aging drugs are primarily targeted at aging-related diseases or pathologies, and not aging itself. Many, in fact, are already pre-existing drugs that may simply be used in an anti-aging function, more or less as a side effect of their normal use. This definitely speeds up the approval process as these drugs have already been shown to be safe. Another way to speed up the process is to bypass it altogether. This is done using so-called nutraceuticals, naturally occurring, therefore non-patentable substances. These are much less regulated. Think supplement aisle in your supermarket. And importantly, since nutraceuticals are substances with regular roles in the body, their mode of action is much more broad, which could be beneficial, for example, a single drug that affects multiple aging mechanisms, or detrimental, such as a much wider range of side effects. To finish up this course, we will talk about the most popular of both pharmaceuticals and nutraceuticals. The most obvious choice of anti-aging drugs are based on one of the oldest mechanistic theories of aging, the free radical theory. If ROS and RNS are fundamental to the aging process, simply scavenging them off should at least be able to alleviate, if not forestall, aging. Naturally occurring antioxidants such as vitamin C or E, or polyphenols from plants, such as green tea catechins or blueberry stilbenoids, have long been thought to have anti-aging effects by scavenging ROS and reducing oxidative damage. Thus has the term superfood been coined to denote plants with particularly high concentrations of various antioxidants. Yet in a situation similar to genetically engineered animals with improved antioxidant capacity, even in a laboratory context, antioxidant supplementation fails to consistently increase longevity. Even an antioxidant, such as vitamin E, which is soluble within the mitochondrial membrane and can directly scavenge ROS as they are produced, does not improve longevity. Yet despite these negative results, antioxidants and especially food or plant-derived nutraceuticals still top the list of promising compounds. Aspirin is another one of those drugs that is firmly rooted in theory, yet with conflicting evidence for efficacy in longevity applications. As we have seen in previous modules, aging is often associated with an increased inflammatory response, so-called inflammaging. Aspirin is a painkiller and anti-inflammatory agent that therefore should be able to treat some of the symptoms of aging. In some animal experiments, aspirin shows not only that it can be used to treat aging-associated phenotypes, but also increase longevity itself. In population studies, aspirin use is associated with increased old age survival. Due to its long usage history, aspirin may in fact be one of the better anti-aging candidates. Another way to treat age-associated damage may be to repair AGE cross-linking products. Another problem associated with AGEs, besides them damaging proteins, is that they can cross-link with each other as well as other proteins, thus forming large, insoluble protein aggregates. This contributes to the functional degeneration, especially of long-lived proteins, such as elastins and collagens in skin and connective tissue. Thus, by breaking AGE cross-links, these proteins may be rejuvenated and age-associated pathologies may be alleviated. One molecule has even made it into phase 2 clinical trials for treating these pathologies, but the effects on longevity are unclear. The realization that insulin signaling, the IIS that we talked about earlier, is involved in longevity regulation has brought anti-diabetic drugs, especially metformin, to the attention of aging researchers. Metformin acts as an anti-diabetic by increasing insulin sensitivity. In animal studies, metformin appears to act as a CR mimetic, reducing methionine metabolism and increasing longevity. Metformin also appears to have some anti-cancer effects, although, as with longevity, these results may be animal strain-dependent. Metformin, as well as other commonly prescribed anti-diabetic medications, thus hold great promise as anti-aging agents as well. Another promising class of potential anti-aging drugs are the sirtuin activating compounds, or STACs. As mentioned earlier, sirtuins are a class of enzymes that perform deacetylation reactions and thus are able to link environmental stimuli to gene activity. Interestingly, sirtuins are dependent on the cofactor NAD+, which signals how much metabolic activity is currently going on in the cell. Sirtuins have been suggested to mediate at least some of the effects of CR, and they may modulate many aspects of metabolism. Sirtuins therefore play a central role in the regulation of aging and longevity. Not surprisingly, therefore, compounds that can activate sirtuins similarly affect health and longevity. The most famous in this regard is resveratrol, a compound found in red grapes and red wine. Resveratrol treatment increases longevity of yeast, worms, and fruit flies, and improves the health of mice fed unhealthy high-fat diets. It moreover appears to act as a DR mimetic. However, resveratrol has large pleiotropic effects, including anti-inflammatory effects, which makes pinpointing its exact mode of action problematic. Nonetheless, due to its anti-disease and anti-aging properties and the fact that there are virtually no side effects. However, let me go on a quick tangent here for a second, if you bear with me. Resveratrol is barely present in white wine, but substantial quantities are present in red wines, especially of the Pinot Noir variety. Some people have calculated that one would have to drink 16 bottles of Pinot Noir per day to get a dose of resveratrol that is similar to what is used in worm longevity studies. Per day. That may be a substantial side effect associated with that. But coming back to my original point, resveratrol and similar compounds are on top of the list for many aging researchers. In fact, some scientists are so convinced of the benefits of resveratrol that they are actually themselves are consuming it. As with IRS or sirtuin signaling, interfering with the third molecular signaling pathway that we have met in this course, the TOR signaling pathway, increases longevity. TOR stands for target of rapamycin, as we've mentioned before, and rapamycin is a drug that was discovered in a bacterium from the Easter island Rapa Nui. Consequently, the protein that rapamycin binds to was called TOR. As we've seen before, the TOR pathway is a nutrient-responsive pathway that is additionally connected to other longevity pathways, such as insulin signaling and the protein quality control pathways. Rapamycin has traditionally been used as an immunosuppressant, especially in organ transplantations, where it is used to inhibit organ rejection. But in the context of aging research, rapamycin inhibits TOR signaling, which increases longevity. In fact, rapamycin thus far is the only pharmaceutical that increases longevity in both male and female mice consistently. Thus, it probably is the most promising anti-aging and longevity drug to date, but due to its immunologic side effect, the practical use of rapamycin may still be limited in a longevity application. Check your understanding. Define nutraceutical and pharmaceutical. Explain tissue replacement rejuvenation as an anti-aging approach. Describe the molecular pathways underlying popular anti-aging treatments, including metformin, Stax, and rapamycin. Discuss the feasibility of anti-aging drugs.